So welcome again. As I mentioned, I'm Frederik Deroose. I will be chairing this session. We will start with a couple of presentations. Today's meeting is all about how to develop and share experiences in target product profiles, of course, specifically for antibiotics. So I've been asking a couple of speakers to give their point of view and also to share their experience from their organization. We have three organizations today represented in this webinar. First, we have Erin Duffy, who is the Chief of R&D of CARB-X. I'm sure that everybody knows Erin. No need for a big introduction. And then it will be followed by, so Erin will give her experience and share the experience from a CARB-X point of view. Then we will move to Francesca Bernardini, who is Vice President at Evotec, who will also share from her own experience, but also from specifically from Evotec's point of view. And then we will move to Gareth Maher-Edwards, who is Scientific Director, Global Health at GSK. So we'll get a perspective from his own experience again, but also, of course, specifically focusing on how GSK is focused on that. The specific element here is that Gareth comes also from a clinical angle, so we'll have today a mix of views that are both in discovery, development, and clinical side of things. So we'll start right away. I see that we have still people coming in. It's okay. That's absolutely fine. And we will start with Erin Duffy. As I mentioned, she will be talking about how CARB-X is expecting a target product profile to be included, for example, in funding applications. I'm sure that some of you or a lot of you have already been involved in funding applications, including CARB-X. And so I will give the floor first to Erin. Erin, if you like, I can do the slides for you. Or I can make you presenter, either way.
Whatever you prefer, Frederik.
Okay, I will give you presenter role.
Okay, perfect.
Okay, so it's maybe a little bit. Just give me a second. Now it should work. And of course, welcome, Erin. I know you're a bit far away in terms of time zones, but thank you anyway for making the time for us. Very much appreciated.
Oh yes, no, hello. Thank you very much for the invitation. I am going to figure out how to make this all work. Let's see. Why can't I'm going to move this somehow? Okay. What are you seeing my presentation now?
Excellent.
Yes. Okay. And it's not cluttered by everything else on the screen.
No. Perfect.
Okay. Okay, well, hello everybody. Good morning, good afternoon, good evening, good night in some cases. As Frederik said, my name is Erin Duffy, and I lead the R&D efforts at CARB-X. So I was asked to talk about TPPs specifically and how we encourage them from applicants, what their purposes and features are. Okay, maybe there's a little bit of a lag. Let's see. Okay, so I think just some general points about TPPs from our perspective. Obviously, and I'll show you a sample TPP from what will be one of the 4 themes in the funding calls that we just launched last week. In our view, the key element here is that target product profiles draw the critical connections among the infection or the syndrome that we're treating, the pathogens associated therewith, and importantly, and I would say evolving certainly in our view, are the performance characteristics that both patients and clinicians need. In our view, TPPs need to be both future looking, and so that's really focused on how one is thinking about the conduct of clinical trials, including what study populations will be targeted. They should also inform a regulatory strategy, and I'll highlight a little of that too. In addition to be future-looking, they should have current relevance. What I mean by that is that, and certainly for the stages of development that we support, the importance is really to influence the design of assay down selection cascade in the discovery phases, so hit to lead, lead optimization of efforts towards product development. And then also to inform the preclinical work streams and resultant packages that are necessary to deliver a clinical candidate that ultimately may be evaluated and approved by a stringent regulatory body. The other important point then of the TPP is to set objective benchmarks for success and progression. So as many of you know, CARB-X funds through various stages of development. So in both therapeutics and prevention, that's anywhere from hit to lead through to a demonstration of safety in humans. And in the case of diagnostics, feasibility through to a demonstration and validation of an alpha prototype. And so it can get a little tricky being a researcher. I certainly was one at some point myself. That as life evolves and that sort of inevitable whittling down of the idea happens, that there are objective benchmarks to bring people back to focus on where you are in terms of meeting the target product profile. Are you continuing to be on success for the ideal target product profile? Have you slipped to what is now a minimal but acceptable target product profile, or has the program unfortunately taken a turn, at some point it's all on the molecule, to a place that isn't going to be attractive for future advanced development, and certainly wouldn't be a competitive product. I think the other thing I want to say, and I think we all know this, but we believe that target product profiles should evolve as the program matures. And when I say that, I really mean program in two senses, both the CARB-X program more generally. So as we learn more, either through internal experience or through interactions with external partners, whether they're downstream developers like BARDA or the AMR Action Fund, GARD-P, or some of our pharmaceutical collaborators, or whether it's other bodies who are setting target product profiles, including, for instance, the WHO. We want to evolve with them so that we have a global view of what the world needs. But then also, as programs progress through our portfolio, certainly we feel that they should evolve as well. So definitely not static. Okay, so this is, if anyone can actually read this, I applaud you, and I apologize for that. It is very tiny, but this is an example target product profile that will go on our website, I believe on Monday, for one of the target product profiles for therapeutics, and that is a lower respiratory tract infection, TPP, focused primarily on LRTIs caused by gram-negative pathogens. And so if you look going down the table here, so in the y-axis, as it were, these are the major categories that we view of a target product profile. So whether that's product indication, the organisms that are covered, the patient population, treatment duration, delivery mode, dosage form, dosage regimen, intended efficacy, risk side effect balance, stability cost, specific population claims, and then a sort of mission statement or value proposition at the very bottom. And so we always look to build both a minimal requirement and an ideal requirement. And again, I'm not going to have you or I read them, but I'm just going to highlight a few things for you. So the first, coming back to this notion of focusing not just on the priority pathogens, but rather the syndromes that they cause and the performance characteristics that are necessary to bring a product to be successful treating them. And so here I've highlighted some things and then I'll blow them out on the right side. So in the ideal target product profile, in this case, we're looking for a therapeutic for hospital associated bacterial pneumonias, as well as ventilator associated bacterial pneumonias. And of course, in terms of the organisms covered, they should be both coverage of sensitive and resistant isolates, including all the relevant genotypes and phenotypes. And here in the ideal case, we look for both activity against Pseudomonas and or Enterobacteriales. So in this case, for instance, it could be Pseudomonas plus Klebsiella, Pseudomonas or Klebsiella as examples. Here we do require both properties, molecular properties that are associated with both oral and IV forms. And that's really important here because what we're looking for is a step down to get people out of the hospital. I must say we just conducted a strategic review, and this was the singular theme in the therapeutics area, which was we need good agents for LRTIs caused by gram-negatives, and we need them to be both oral and IV. In terms of the safety proposition, you know, there's this sort of general feeling of manageable AEs with at least a 5x safety window or therapeutic margin. And then finally here, we would seek to have cost of goods that would enable a low middle income country launch. And I say that in part because from the strategic review, it was very clear to us that the pathogen set differs. If you think about high income countries versus high income countries plus low middle income countries or LMICs as well, where I'll just say that in high income countries, what we often heard was, I don't even know what a Klebsiella pneumonia looks like, to clinicians in low middle income countries saying that's our biggest problem. And so we need here in this case to be open to both. Okay, on the other side of the coin then, for the minimal TPP, again, focused on HAP-VAP. Oh, and I should have said a couple of things here written in the bottom. That certainly nice to have in this ideal TPP would be the inclusion of Acinetobacter baumannii and or expansion to hospitalized community-acquired bacterial pneumonia, and of course the associated pathogens associated therewith, which again, very different set that would now bring in your gram-positives as well as your fastidious gram-negatives. But again, all with then that link to an oral form. Thinking about a minimal TPP, then we think about HAP-VAP exclusively, and again, sensitive and resistant isolates now where Pseudomonas is required plus minus the Enterobacteriales. And here IV form is acceptable, so we're really focused on you know, serious hospital-based infections, but again, where Pseudomonas then is a primary key. Here again, manageable AEs, but given to the serious nature of these infections, a therapeutic window of 3 or more. And here then, in terms of availability, we look for products to be equivalent to current treatment regimens in high income countries. And then finally, again, a buy-up or a nice to have would include Acinetobacter baumannii. So just to sum in terms of this triad of syndromes, pathogens, and performance, really does focus us then and our product developers on the microbiology and pharmacology covering the relevant pathogens for the infection type. And of course, this needs to be informed, as I highlighted a little earlier about Klebsiella by current epidemiology. It also emphasizes where the drug needs to be, and then finally keeps an eye on trades. What I mean by that is as I mentioned earlier, unfortunately, we've all experienced it where as program goes on and you learn more about the molecule, things start to erode, and we just want to make sure that we understand as things start to come off the table what kind of product we have. So again, if all of a sudden we recognize that an oral option just isn't possible given the molecular space where we've landed, then that really changes the calculus on what the coverage must be. And in that case, then what we avoid is that pitfall of not another IV UTI drug that comes out of the pot. Okay, so then just to highlight one other area here, which would be the clinical and regulatory implications. So I've highlighted a few words and then bringing them out here in the ideal case. So here then we would be focused on pneumonias, and so one might think about and queue up then a BAL study, a phase 1 study that might be timed very closely to the first in human study, just to get a really good sense of is the drug showing up for its job like it had preclinically. Turns patient population, adults in a healthcare setting. Now, in this case, in the ideal case, we'd like to cover children too. Often people get a waiver and kick that can down the road. But if we're focused on children here, then how do we front load that strategy? And do we engage partners? For instance, GARD-P is very active in this space. So again, helping people to plan for what you need early on. In terms of what we need for efficacy, then in this case focused on either non-inferiority or superiority design. And in this scenario, then what would those designs be? What would endpoints be? And to think about these early, and then to think about what supportive non-clinical data we need to make those designs most effective. And then finally, in terms of safety, very much listening to what happens preclinically, and then how and when to sequence additional phase 1s to underscore as appropriate clinical proof that safety, both safety pharmacology and toxicology, are underscored. Likewise, then in the minimal case, similarly, again, the cases here, pneumonia, now focus squarely on adults where NI is perfectly acceptable. And then again, when do you sequence those phase 1s and what do they need to be? Okay, so that is it. I want to say, though, that we will be publishing on Monday many things on our website, including the target product profiles for all 4 of our funding themes. I know this is a little promotional, and I'm at my time, but just to say, if you missed us in Basel last week, we did announce 4 funding themes, from the therapeutic that I just talked about for infections caused by gram-negative pathogens, through to a prevention theme for disseminated or invasive disease of Staph aureus or E. coli, and then 2 diagnostic calls, one product focus for neonatal sepsis, and one a little upstream from that, which is to focus on novel sample types for lower respiratory tract infections. So again, we'll be publishing target product profiles for all of these ahead of opening the portal for the funding call. There will be 2 cycles, and here are the dates, but again, this will all be on our website, and we do hope you join us in these calls. So thank you very much, and I look forward to the discussion.
Thank you so much, Erin. And it's absolutely fine to do a little promotion. It's absolutely, I think many people appreciate, and not everybody was last week in the AMR conference indeed. So thank you very much. We'll keep the questions till the end, and we'll have a panel discussion as well. But yeah, thank you so much for sharing your thoughts with us, and very practical examples. So really great. So let's move to the next speaker. Is Francesca from Evotec, who will give her perspective from her organization. Just a little introduction on yourself, Francesca. So Francesca graduated in chemistry and got her PhD in biochemical and pharmaceutical methodologies. She has over 20 years of research experience in an international context, mostly in pharmaceutical biotech companies such as Arpida, Polyfor, Debiopharm, working on antibiotic research and preclinical development. She has covered different roles, such as microbiology team leader, project leader, and preclinical operations lead. Francesca joined Evotec in February 2021 to lead the Lyon's IVB department activities, microbial programs. She also covers the role of AMR therapeutic area lead to support partnered projects. Very impressive CV, Francesca. Welcome. And would you like to do the slides as well? Shall I make you presenter?
If you can present them, I would appreciate. Thanks a lot.
Okay. So welcome, and just give me a second.
So in the meantime, thanks a lot, Frederik, for your very kind introduction, and welcome to everyone to this webinar. For those of you who are not very familiar with Evotec detail, we are a life science partnering company, meaning that we conduct a wide variety of modality agnostic drug discovery programs with different partners across a range of therapeutic area, including antimicrobial resistance. As I'm sure is true for everyone listening today, we aim to address areas of high unmet medical need by bringing all possible innovation to the table, but also conducting projects in the most efficient manner in order to deliver projects with the greatest chance of success. And the need for innovation and efficiency is particularly acute in the field of IMR. To deliver with this scope, we found that to develop and continuously use TPP is critical throughout the drug discovery process all along, and to guide the whole process from the initial idea to the identification of the clinical candidate. If you could please move to the first slide. The next one, please. So because we work in partnership, we develop TPPs together with our partners, and this is key to get on the same page from the very beginning of the project regarding what we are striving for together, to ensure that we have a tool to evaluate where we are compared to our vision and to enable rapid, clear decision-making based on solid set of data. So we acknowledge that the TPP is only a tool, and which one we want to pursue can be changed, but this is a critical tool for setting up this clinical scale that identify the activities and models needed to address all the critical points to be taken into account in the multi-parametric optimization which take place from high-throughput screening to the lead optimization phase until the final clinical candidate selection. It provides clear guidance throughout the drug discovery, but at the same time, it's critical to keep some flexibility both at the start of the program, when the information on series is rather limited, and throughout the project progression to exploit most of the potential of any new chemical series. However, at Evotec, it is more than that. We have been working on an amazingly increasing number of projects using new modalities or addressing non-traditional targets. And for these new approaches, the MPK, CMC, and safety may represent unexplored avenues and then new challenges. At the same time, however, new opportunities. So we use also the TPP to identify relevant translational model to develop, for example, using clinical isolates from seriously ill patients, or as another example, using cystic fibrosis isolates to set up a lung infection model relevant for this kind of disease. We can also qualify models on the basis of host infection biomarkers, which could be particularly relevant, for example, if a host target therapy is envisaged. And of course, new modalities may also require to set up adapted in vivo translational models. So if I may summarize, at each step, the TPP will be used as support for gap analysis to drive the next steps of the projects and will help in making decisions in a timely manner. If you could please move to the next slide. So the TPP, of course, needs to be developed as early as possible. And at Evotec, typically during drug discovery in a hit-to-lead phase, we would focus on the spectrum of action of the compounds, their coverage of clinically relevant isolates, and on physicochemical and ADME properties of the compound. Once the lead series has been identified, we would focus our activity on a series mode of action, especially if the target is unknown. We would de-risk the project from development of resistance point of view. We would aim at gathering preliminary safety, and then, of course, focus a lot on PK/PD package in order to have a solid set of data that would be used for human efficacious dose prediction. Of course, then the question may come, how narrow or broad the TPP should be, how flexible or how fixed it should be in the discovery phase. So to de-risk a program and increase chances of success, I would answer that the choice of possible TPP should be as broad as possible, including, as mentioned by Erin, whenever possible, a base minimal case and a preferred, optimal, or desired case. However, the associated selection criteria should be as stringent as possible. Flexibility in the discovery phase is critical to avoid discarding any promising new chemical matter or modality. However, if the series does not fulfill the criteria to give the product with the desired TPP characteristics, the choice should be made as soon as possible to target a different TPP. Sometimes this may come as early, already in the hit to lead phase based on the spectrum, or later on in the lead optimization phase when data of efficacy and pharmacological properties of the series are available. But that still focusing on the drug discovery process. Of course, other information will be available along the project progression, and change of TPP may eventually come even later beyond the preclinical phase. But I can illustrate that with a showcase. So if you could please move to the next slide, Frederik. So in this example, the project team made a literature search, and they identified a series of small molecules very easily available by synthesis and with the likelihood to be developed for an oral administration. Based on the literature, the series had the potential of becoming either a gram-negative antibiotic or a gram-positive antibiotic, and apparently there was not much, the series had not been exploited so much. So for us, it was represented at an ideal starting point. The team identified for this series two TPP, TPP1, where both gram-negative and gram-positive activity were taken into consideration. So a compound eventually having only gram-negative activity would be considered as a base case TPP, and an antibiotic covering both gram-negative and gram-positive would be the ideal TPP. TPP2, focus on gram-positive activity, was also identified. And in these two TPP, the primary indication had been discussed. As a proof of concept in the clinics, a complicated urinary tract infection treatment was envisaged for the TPP1, and acute bacterial skin structure, and soft tissues infection had been envisaged as clinical proof of concept for a TPP2. The spectrum has been described for both TPP. As order of entry, the series would be positioned as first in class, exploiting a new mode of action, and also efficacy, safety were decided or described, and at the same time, administration route and duration of the treatment. As a possibility, a third TPP, rather single pathogen TPP, had also been envisaged, and in particular, Neisseria gonorrhoeae was identified as the ideal target for this kind of narrow spectrum antibiotic. If you could please move to the next slide. In the lead optimization, in data expansion phase, we first focus on gram-negative activity because this is the major unmet medical need. And as you can see here from the data, the compounds showed very promising activity against both E. coli and Acinetobacter baumannii, which are in the top list priority of escape pathogen to treat. However, the activity against Klebsiella was less potent, and there was no coverage for Pseudomonas. In the meantime, we also got an alert of resistance development against both E. coli and Acinetobacter. And based on one hand on the limited spectrum and the high risk of development of resistance, this TPP has been deprioritized in favor of TPP focusing on gram-positive activity. So if you could please go to the next slide. So the best compounds show very promising activity against Staphylococcus aureus in a panel which includes multi-drug resistant isolates, including methicillin resistant. The MICs were pretty good with value between 2 and 4 against this panel. The activity against the enterococci was weaker or absent, and also the coverage of Streptococcus pneumonia was rather limited. So this would indicate that in this case, a base case gram-positive TPP could be envisaged covering mainly Staphylococcus aureus, against which the risk of development of resistance is very low, we had very promising frequency of resistance values for these species. And that, beside the potential of an oral route as way of administering the compounds, makes this series, of course, quite appealing for this kind of application. However, in parallel, I would say almost by serendipity, the compounds were profiled for their activity against Neisseria. And if you could move to the next slide, please. Bingo, the compounds are very potent against a very nasty panel of Neisseria gonorrhoeae, including ceftriaxone resistant isolates. Some of them are also resistant to ciprofloxacin. The best compounds show MICs equal or below 0.125 microgram per ml. The best MIC90 are as low as 0.25 microgram per ml. And also for this species, the frequency of resistance are in the range of 10 to the minus 10. Hence, after very limited heat expansion, we are in the position now to decide that the targeted TPP would be the TPP3, addressing single pathogen agent to treat Neisseria gonorrhoeae. So if I may summarize what is our vision of TPP. So the TPP is the frame to drive the drug discovery and to facilitate efficient and clear decision making. However, it's only a tool, and the choice of which TPP will be finally addressed should be as broad and as flexible as possible to increase the probability of success of the project. However, the associated selection criteria should be as stringent as possible to make sure that the final clinical candidate selection will give a product with the best great chances of success to become a treatment for an unmet medical need. And with that, I would like to thank you for your attention. And thanks, Frederik, for changing the slide for me.
No worries, no worries. Happy that it worked nicely. Thank you so much, Francesca, and also, again, also as Erin did, for being very practical and giving these real-life examples. It's highly appreciated. I'm sure there will be some questions. As I said, we'll do that after the presentation. So please stay with us, and we'll come back to you with some questions from the audience. Okay. So the next speaker, the third speaker, is Gareth Maher-Edwards, and Edwards, same question for you. Would you like me to do your slides, or would you want to make presenter?
Yeah,
or
if you can drive them, given I've had challenges to sort volume out here, that might be better. Yeah, so no, just concentrate on speaking. Thanks. Yeah.
Yes, yes. All right. Why don't I first introduce you to the audience? So welcome, Gareth. So Gareth has approximately 30 years of experience across all stages of clinical development in a variety of therapeutic areas. Currently, Gareth is Scientific Director in Global Health Medicines R&D at GSK, where he is the medicine development leader for a TB asset, which is currently in phase 2 development, and also leads the project for another TB asset currently in phase 1. So as I mentioned in the introduction, Gareth is also going to talk, first of all, also about TPP development from a clinical perspective and also from a GSK perspective. So welcome, Gareth, and I will do the slides for you.
So just while you bring those up, then, I mean, thank you for the invitation to take part in this webinar. I think it's a really great topic, and I think it's great to get such a broad audience to kind of debate this and you know, see how we kind of join up with our thinking around this. So thank you very much for that. Okay, if you want to move on to the next slide. So just very quickly, disclosures. Obviously, I'm a full-time employee of GSK, so I'm paid by GSK, and I have shares in GSK. The content of the presentation, I will talk about how we approach TPPs in GSK. Some of my commentary might be my own personal opinions rather than necessarily, from my experience, rather than necessarily a reflection of GSK policy. Next slide. So I'd like to start with, perspective around what is a TPP and why do we have them. So on the left of the slide here, I just copied out actually the WHO definition of a TPP, which is something that outlines the desired profile characteristics of a target product that's aimed at a particular disease or set of diseases. And I think, my definition, our definition, GSK, pretty much well aligned with that. Similarly to that's been discussed by the other speakers, these will include information around what's the target population, the safety and efficacy requirements of the product, and other important attributes or characteristics of the product that will hopefully frame up what will make for a successful medicine. So why do we have these? So really they set the bar for defining what is likely to make for a differentiated and reimbursable medicine that's going to make a difference for patients. So really, I think within GSK, we see the target product profile very much as our North Star for our entire development strategy of a product. It provides the basis for generation of an integrated evidence plan, and that will include the clinical development plan, but also considerations around kind of health economic data and other types of data that will be supportive around access and not just registration, and also lots of information around, what's the formulation of the product need to look like, how can this be delivered into the settings where the patients are there needs for companion diagnostics and other such really important considerations. And with this, we can guide the advancement of the asset through development, or when necessary, we've generated a set of thresholds that then enable us to consider when we should perhaps stop developing the product. So this, in a way, it kind of gives us an anchor. It's a bit of a lighthouse, a beacon. It keeps us away from paths that we don't want to go down, and it sets our direction for the forward and future development of the project. To move to the next slide. So this is kind of a blank template of what a TPP might look like in GSK. Again, we have the indication target populations, and we might capture some information around what the anticipated standard of cares will be at launch, and then we'll start to capture information around what is the unmet need and the key claims that will drive value for this particular indication. So this is kind of the why and the how of the target profile, and it might address such questions as what are the challenges or unmet needs that this program or this asset needs to address. Is this going to be something that is a first time approval for the disease, or is the market already quite crowded with current treatments? Is this medicine going to define, maybe if it is a first entry into a particular disease space, or can significantly shift the treatment paradigm? And where does this medicine sit with respect to current standards of care, but also perhaps other things that might be in development? And is there a key patient population within the overall indication that's absolutely critical to driving the value for patients? And like the other speakers have talked about, when we go through the kind of characteristics and profile of our assets and the target profile that we're looking for, we split this into a minimal, a target, and an optimistic profile. The minimum profile really represents the threshold below which the product wouldn't be launched or would not be further developed. So that really is your kind of, in a way, your stop criteria. The target profile is what we aim for. So this represents where we're aiming to get to, and is based on what we consider would enable us to have a differentiated and reimbursable medicine within the space that we're working in. It's aspirational, but it's also backed by credible assumptions and data. And early in development, that might be framed mostly by the biology or genetics, and over time it will shift more to the clinical evidence that's being developed. Our optimistic profile, so that represents a profile that would deliver significant incremental value beyond the target ambitions. So, some properties that might drive to a greater market share or increased access to patient populations. And generally, this will obviously have a lower probability of success, but still needs to be something that's, to some extent realistic. And we'll also capture then key drivers of success. So this could be things like thinking about the time to market entry, where that asset would be in the order of entry to the market, considerations about pricing and cost of goods, IP considerations, thoughts about the patient population, how accessible are they? Are companion diagnostics going to be required to identify the patient population? Competitive considerations, thoughts about dosing, need for patient-reported outcomes or health outcome data, and maybe other populations such as pediatrics. If you want to move to the next slide. So this is very similar to a slide actually that Francesca showed. So obviously I tend to work more towards the right-hand side of this slide, but the question here is, when do you need a TPP? And back to what I said at the start, it's our North Star. This is everything that defines what we need to work towards. Right from the start, we need a TPP. We need to know where we're trying to get to before we even start with anything, including target validation. So how is it useful along this journey? So at target validation, it helps us to think about whether the targets we're looking at match where we're trying to get to in terms of the disease or the patient population. Lead discovery, lead optimization, which Francesca talked about already, are the candidates we're looking at likely to match the intended use? And we might have a significant focus here on candidate quality criteria. Is the quality of the candidate going to be good enough to be used as a medicine in this population? Preclinical, we want to start understanding if the asset is demonstrating, at least in preclinical models, efficacy, PK characteristics and safety that is going to be translatable to the clinic and in line with the TPP. In phase 1, we start to look at that for a healthy volunteer human setting, and obviously into phase 2. We're starting to try and drive some signals within patients that tell us, hopefully, not just that there's some effect of the drug, but that effect of the drug is something that we can link to a likely outcome that meets the TPP requirements. We can also start to think around how the dosing regimen fits with TPP requirements and early indications around safety. We can further characterize that through phase 3, and also in phase 3 really start to generate those data around whether we're having a profile of drug that's meeting those key drivers. If we can start to identify those things around the key drivers earlier in phase 2, then that obviously is hugely advantageous for the significant investment decisions that we need to go into phase 3. And throughout this process, we're really trying to continually ask ourselves the question as to whether the profile of this drug is meeting that TPP to address an unmet need. What's the advantage of this going to be? How does it differentiate from other treatments that are already out there for a particular disease? We'll be looking externally, are there guidances from external regulatory, WHO, other important bodies within the disease space that help to guide what the profile needs to look like, and thinking about how this might differ across different patient segments, maybe across different countries and regions. Next slide. So what inputs and drivers determine the TPP? So a lot of this comes from external sources, really. So other regulatory requirements or guidance, but also what are payers' reimbursement access? What are the needs there? How does that impact the profile that we need to look for? Prescribers, what do they need? Patients, what's the patient need? And are there advocacy or other stakeholders around the patient space that are helpful to inform what the profile of a new drug should look like in that disease space? Obviously, our internal pipeline and a competitor landscape is important in terms of driving the profile that we'll look for, and always looking for those differentiating factors for that particular asset. Next slide. So what are some of the benefits of a clear and focused TPP? So primarily, obviously, the bias of my career in clinical development, it's first and foremost of benefit to the project or product development team, because it sets the direction of travel and the targets, and it sets the aims out very clearly. It completely informs study design and the clinical development plan, and determines the evidence requirements that need to come from that. It helps to think about the right populations to include, when and where you might need to generate active comparator data, and the value of things like patient-reported outcomes and health economics. It's beneficial for senior management and governance. It creates for them a benchmark for success within a portfolio of drugs for a particular disease, and that really helps with decision making and obviously picking the winners within any portfolio. The two benefits of that really are, one, any progression decision can be made with real confidence that based on the profile that you're seeing, it's going to meet an unmet need that has a likelihood to make its way to patients, but it also helps with really timely termination. And that has the benefit of enabling resources to be diverted to something that's more likely to reach a profile that's going to match that unmet need and make its way to patients. Within global health in GSK, everything we do in global health, we do in collaboration with other partners. Global health is a resource constrained space, and so pulling together resources is hugely important for success in that space. And having a clear TPP really helps with those collaborations, because it enables us to be very, very clear about what our strategy is and where we're trying to drive towards in terms of any particular product that we're developing. And that's equally useful for regulatories and any other external interactions, because it enables us to clearly describe what the potential benefits of the asset are, what differentiation that has from current treatments, and contextualize the scientific rationale around the product. Next slide, which I think is my last slide. So yeah, so how do we keep it current and relevant? So it's defined in GSK. We set our TPPs, or the way our TPPs need to be set up based on a disease area strategy. But the details of that TPP is very much a team effort from various subject matter experts around the organisation. That includes physicians and scientists, but it includes input from safety groups, regulatory, access, within kind of non-global health settings, obviously from commercial colleagues, local input from our LOCs where we have them, and their relevant regions for the particular medicine, clinical operations statistics, and many, many others. But we test and validate these TPPs externally so that we really understand that it's not just an internal thought process, but there's a buy-in to what we're trying to do externally around feasibility, the reality on the ground, and the ability to gain access to patients and really create impact. And we might do this through advisory boards or scientific workshops, or commissioning market research or feasibility studies. And then we'll also do research around things like both the cost effectiveness of the product, but also a kind of willingness to pay. So we might put a profile out there in front of kind of national programs or whatever, and see whether that is a profile that, based on the resources and budget available in a country, would actually be something that might be purchased for use within that disease space. And we can also do impact modelling, which helps us to understand whether that profile is going to lead to a significant impact in the patient space. So I think that's my time up, and my overview of how we use the TPPs within GSK.
Yes, thank you very much, Gareth. And it's very nice to see that there's of course alignment and some overlapping in the three presentations, luckily, but there's also some specific elements that you brought up from a GSK point of view, but also from the clinical point of view. So highly appreciate the mix that we have. And also to the audience, when we asked you to register, was also on a purpose, of course, to give you the meeting details, but also what we analyzed is that the background of people is different, and also the diversity of people that we have in this webinar is also people from early discovery phases all the way to clinical. So we have different types of organizations on the line. So, and not everybody is, of course, aware of all the aspects of a TPP. So definitely thank you, Francesca and Erin and Gareth, for giving this level of insight. What we're now going to do, can I maybe ask Erin also to switch on your video if you can? And Lena, we don't, so we don't, yeah, I just want to have 3 speakers and no, well, hi Lena, by the way.
Yes, hello. Sorry, I just clicked on the link here.
No problem,
and I was
no
late
problem,
to the webinar. Maybe you're already.
No problem. You can switch off your video, Lena. That's fine.
Yeah.
So what we're going to do next, and Clément, if you can also help me a little bit with this, is as we mentioned in the beginning, we're gonna, it might be a little bit more of a challenge. We're gonna start a panel discussion, and this is not like in a real life conference, but it's on a video. So forgive me if maybe some people that we missed some of the questions. So we will follow up. We noted them down, and also have some additional questions. And so this is actually the fun part, I hope, for you, Erin, Francesca, and Gareth, is that we will do a small panel discussion, and I hope there will be different views. Maybe, Clément, we can start with some of the questions of the audience, and then I can also add some more questions that I prepared. So, but let's give the priority to the audience.
Yes, absolutely. I hope you can hear me well. So far, there are 4 questions posted in the chat, 3 for Erin and 1 for Francesca. So let's just start in the chronological order. And the first question for Erin is about the scope for an IV agent against Acinetobacter baumannii infections. So if you could please, Erin, clarify what the scope is.
Yeah, so again, for us, that would not be a standalone priority. So it would be added on either to the ideal or the minimal TPP as I outlined there. So again, either, coverage of Pseudomonas plus the Enterobacteriales plus Acinetobacter, or one of Pseudomonas or the Enterobacteriales and Acinetobacter, but then all the other profiles would stay the same. I do note, I know we're not allowed to call on the audience, but I did see Alita Miller here who probably could wax philosophically about, what it takes to develop an Acinetobacter drug given their recent success.
Okay, thanks, Erin. I hope this answers the questions for Manoj. If not, please feel free to post the follow-on questions in the chat. Then there's a second question from either Fairhead, which I'm not sure I entirely understand clearly. The question is, what proof of concept study would be possible for Pseudomonas aeruginosa ventilator-associated pneumonia, given the product will be tested in combination with standard of care versus standard of care making a very large study to show a difference. If you need clarification on this question, Erin, I don't need to ask, and either can unmute herself.
Yeah, no, and I certainly think I understand it. I'd be interested in my fellow panelists' view on this. But one thing that I neglected to say that we've done at CARB-X, and we've started to do this as product developers come to the end of lead optimization, where you're declaring a clinical candidate and you're about to set off, on an IND or other regulatory enabling path. Is to nucleate a clinical advisory board. And part of that is, again, working with the TPP and thinking about your label and what you need to do, working back through what data do you have that support the activity of your agent, and how that then would relate to, what evidence you need to amass to be confident before you start, tripping that trigger of pretty significant spend in the clinic. And so we often differentiate the term proof of concept, which is more of the sort of classic phase 2 dress rehearsal, as it were, for what you're going to do in your pivotal study. to proof of mechanism, which is a little different. And the degree to which you might have to do clinical proof of mechanism very much, I think, depends on what the preclinical package that you have. And again, working back from your TPPs. So, what PK/PD package do you have that really demonstrates robustness in an efficacy response in animals? What underscoring of mechanism do you have in terms of your target and your activity and your engagement? And then, if you don't, with traditional small molecule antibiotics, that may be one thing. For non-traditionals, which is a little bit of a different flavor here, that may be something else, where then that proof of mechanism may need to be in the clinic. It may be a different kind of study, not along the path for your ultimate goal of HAP-VAP, but what kind of study could you do to demonstrate Pseudomonas engagement? Is it something like burns or wounds or diabetic foot, or what could you envision that you could do in a small setting to underscore proof of mechanism mechanism ahead of jumping into your pivotal pneumonia studies? So just some thoughts there. And again, love to hear from the other panelists how you think about things like this.
Yeah, thanks, Erin, for this insight. Francesca, you want to comment as well?
Maybe I can comment because I'm coming from my previous experience in developing muripavidine, a Pseudomonas-specific antibiotic, indeed, which was intended to be developed for treatment of HAP-VAP. As a preliminary proof of concept, what was envisaged at that time was treatment of non-CF bronchiectasis. However, this was pursued in parallel to the phase 2 in HAP-VAP, which was then continued, and the phase 3 was indeed envisaged. And clearly, the fact that you need to demonstrate superior efficiency in combination with the standard of care compared to the standard of care itself implies a very large number of patients, a large number of clinical sites, and puts the hurdle quite significant. And this outline, I would say, the need to have contact with regulatory bodies as early as possible in the project, because they will be the ultimate approver of the investigator brochure to have the first in human proof of concept, and then later on for the marketing of the drug. And if they would not accept what has been proposed, basically it may lead to a failure of the project. And then when you envisage also an antibiotics and pathogen-specific antibiotic, then of course an additional hurdle may be the need of early diagnostics to be sure that you select the right population of patients to treat. And we have to keep in mind that for Pseudomonas, the mortality associated to this pathogen when induces HAP-VAP is very high. However, the incidence is luckily relatively low. So all these aspects, of course, need to be taken into account.
Thanks, Francesca. I don't know, Gareth, if you want to add something more.
I mean, I don't have anything specific to add, but I think in a general sense with proof of concept, it's always a challenging space. I think, obviously the ideal is to design a study that exposes the fewest participants for the shortest space of time at the lowest cost that gives you the clearest answer, but that's not always that easy to do, as the other speakers have illustrated. And then I think from a practical point of view, then it often comes down to a consideration of where you want to draw the line with your risk, because there's always a risk from any study around a false positive and a false negative, and you have to consider where, based on the space you're working in, you're prepared to draw that risk. Are you happier to take the risk of moving something forward that perhaps is going to fail at the next stage, or are you is the space a very challenging one where you want to set the bar higher, and you accept with that there's a risk of potentially discarding assets that actually might have been useful?
Absolutely, fully agree. Thanks, the three of you. We have a mix of more broader questions and very specific ones, so we're going to have a mix of it. So the next one, Clément, I can take this one, is for Francesca on the proof of principle for the example that you gave. So I think the question is actually which infection model would be your first choice for POP in the model that you gave. I think there was a natural product program that you gave as a specific example. So any advice on the infection models?
For sure, working on this area is maybe a challenge because it's a difficult pathogen to handle, and to have stable murine model to prove the efficacy is not that straightforward. At Evotec, we won a model of vaginal infection with Neisseria, and this is what will be used for the proof of concept, or a preliminary proof of concept for this pathogen. Then an alternative when the in vivo murine model choice is limited could be to test the efficacy using the hollow fiber approach, which allows to test, I would say, larger number of clinical isolates to have proof of efficacy first, but also to assess what would be the potential of resistance development against this pathogen. And this is something that we're also developing.
Yeah, thank you very much, Francesca.
Yeah.
I'm not sure if we still have, do we still have Erin?
You do. I'm having troubles with my camera. It keeps
the
coming
camera
on
is
and
fine.
coming off. Sorry.
It's fine.
Okay.
It's fine. No worries. I have a question for you. Well, from the audience. I think it's also very interesting. All questions, by the way, are interesting. But so at what stage of the development does your, as you were talking about, a 3 to 5-fold therapeutic window, therapeutic index apply? Does it apply to all stages, or were you referring to a specific stage? So, I mean, that's the question from Derry Mercer.
Yeah, I mean, that's a great question. And this is where I think the sort of interplay of the TPP and your down selection, cascade become very important. But, I think where we start to think about that number is really in that once you have a lead or set of leads where now you have a reasonable sense of what, efficacy looks like. Maybe you don't have all of the studies in pathogens and, that you want, but you've done a few models, you have a sense of where that sweet spot is for efficacy, and you've done some, non-GLP, safety study, maybe like a 5-day rat study or something where you understand, what the, at least the early clinical signs and concentrations or exposures associated therewith. And that's kind of the first time that we think about when you can look at, what a window might be. And then as you advance, obviously that becomes, more clear.
Thank you, Erin.
Is that helpful?
I think so. There was also a specific follow-up question on Manoj on the CARB-X application. I would say that we take this one offline. If there are specific questions around a certain call for a grant, if you don't mind, I will also, I can send you the question, but I think this is something specific related to the call. And so let's move to the next question. I think this is also for the 3 speakers. So in the ideal, we talk about different types of TPPs, minimal, ideal, and preferred, and sort of the different terminologies that you used. In the ideal TPP, the oral step down was mentioned, right? So I think by the 3 of you. The question is, how important is this for a preclinical project with a focus for patients in hospital settings? I think this is an easy question, but maybe a more difficult thing to address. So, but maybe, Erin, I can first go to Gareth, and then we'll
yeah, absolutely.
Gareth.
So it was the question was, what's the importance of an oral?
Right. So how important is this for, well, if you, and what do you need to take into considerations in your preclinical project where it all starts and your TPP evolves, indeed with a focus for patients in a hospital setting?
Yeah, so I'm probably not the best person to answer that, but I mean, I think it depends very much on the setting. I think whether, an oral route of delivery is crucially important or not, but I guess there's more than one factor to consider. So there's what the impact of that is for application delivery for the patient, but there's also what the importance of that is for target engagement, and actually getting activity of the drug, and there's a balance to be made between those two items. But I guess within a hospital setting, from an uneducated point of view, it's possibly from a practical point of view less of a consideration. In an outpatient setting where, you know, traditionally that's where we're looking at in the TB space, it's clearly really important that we've got oral medications that are easier for the patient use.
Thank you, Gareth. Erin, I think you want to comment on this as well.
Yeah, so again, for us, you know, the oral form really has two drivers. One is about access at various levels of the healthcare system. So having an oral option. Now, obviously, if you're on a ventilator, the oral doesn't matter right away. But in that scenario, then you're hoping to start with an IV, but then have that oral option to transition people out. But specifically to the question about when do you worry about it, you better worry about it early, because, you know, once the properties of the molecule are baked in, you know, this notion that like a prodrug pixie dust is going to save everything is, you know, frankly, sometimes miracles happen, but usually they don't. And so the sooner you think about the property set that drives the oral activity, if that's part of the TPP, and then you carry that through as part of your multifactorial design, the better off you're going to be.
Yeah, absolutely. Francesca, you want to add something?
I would say that after the comments of my colleagues, there is not so much to add on this point. However, I agree that the option to include an oral step down should be in the TPP since the beginning. Whether this is possible, maybe for hospital setting would not be the most critical point. Then it would depend, of course, which kind of infection we want to treat. In the example that I gave, Neisseria, there an oral option would be the preferred, but we have also to keep in mind the patients are not in the, say, at least they should not be in intensive care unit. If they are, they will not be for Neisseria infection. Just as a complementary comment.
Absolutely, absolutely. Gareth, we're going back to you. The next one is for you. So you touched upon this, but there was a little bit, if you can elaborate a bit more on the following. What are the commercial implications? You mentioned associated market size, drug pricing potential of these unmet need-driven TPPs. Are there certain TPPs that are more commercially favorable than others? I think you gave some examples, but can you elaborate a bit more from your own perspective or from GSK, that doesn't matter.
Yeah, and I think this is a great question. It's often quite a difficult. I have a colleague who talks about squaring circles, so it's often a difficult circle to square, I think, trying to balance these things. So yeah, so in respect of unmet need and market size, you can often find that actually these things can be inversely proportional, and I think TB is a really great example of that. You know, the greatest unmet need in TB is in people with significant levels of drug resistance, but that's a much smaller portion of the overall TB population compared to drug susceptible TB. And we've seen that in other areas and sectors that can influence the price of a product. So in rare diseases can often command a reasonably high price because there's massively significant unmet need. There's not necessarily a lot of alternative treatments. But in the area of global health, when we're talking about making medicines available in low income and low middle income countries, the ability to influence or dictate prices are much more reduced, because at the end of the day, the medicine simply has to be affordable to reach patients. And so within the TPPs that we will generate within TB, cost is absolutely crucial, and it's really important that we can generate a new medicine within the kind of guidelines that are set by the WHO, and so cost of goods becomes really crucially important. Market size, as I said, it is important, generally speaking. A larger market size from a commercial perspective, if you like, or even just from thinking about a global health impact perspective, the better that can be. But again, I think there's always a depth and breadth argument about unmet need. So you can think about unmet need in terms of how many patients can you treat, and you could treat millions of patients with a fairly marginal impact to most of that population in terms of their outcomes, or you could focus in on a smaller segment of that population, and actually for each individual patient your impact on their outcome is far more significant, because without the new intervention, their prognosis could be much more considerably negative. So there's kind of a balance to these things that's not necessarily easy to find the right point of that. But these are really, really important considerations in the way that we construct our TPP and the way that we think about what we progress and what we don't progress through development.
Thank you so much, Gareth. Francesca, Erin, anything you would like to add to this question, or is it okay? Thanks, Gareth. I'm just cautious of the time. We only have 10-15 minutes left, so we're not going to be able to cover all the questions. So, but that's absolutely fine. I'm very happy that we received so many questions. So, I'm just going to see the kind of questions that might already be addressed, at least by some of you. But let's just see for completion that we have this. I think, Gareth, you mentioned, so on the next question is, which kind of experts do you involve in your organization to develop TPPs? I think you had a slide on that, and it is quite a number of people that are involved in this. So I think you addressed this, Gareth, but I'm just maybe from a CARB-X point of view. Erin, it's a different type of organization. It's a funding organization. So maybe you can also address a bit on from your point of view, what kind of experts you involve and in your organization to develop these TPPs, maybe from a different angle.
Yeah, thank you, Frederik. So for us, it's takes a village, I guess, is what I would say. So we, as I think I might have mentioned, but we have recently completed a refresh of a strategic review, which is top to bottom, to come to the funding themes that we elevated a week ago for funding. And these are panels of about 10 to 12 experts, largely ID clinicians, globally distributed. Again, our goal is not only to produce products for high-income countries, but also low middle income countries as well. And we had panels actually for each pillar, as it were, so therapeutics, prevention, and diagnosis. But in addition to that, once those concepts were solidified and product characteristics were discussed, we also then engage experts within our subject matter network, which is about 250 people or so, largely former pharma and biotech practitioners in the space, who help us to think about and refine the TPPs. But also, we are very earnest about working with our downstream partners. I mentioned like BARDA, the AMR Action Fund, GARD-P, and some of the pharmas that remain active in the space, because we don't want to be building products that they don't want to embrace and take from where we end, which is end of first in human, and through to approval. So really need buy-in from all of those groups. And then finally, other bodies, including and importantly, the WHO, who have really taken a leadership position in the space. So we want to make sure that whatever we do is harmonized, so that, again, we have the best global TPPs that are possible. I want to say one other thing, though, which is that these are living documents in our view, too. And so we learn, and as we learn, they evolve.
Thank you, Erin. Francesca, I'm going to give you another question, because I'm sure that at Evotec you also have a lot of people that you involve, and I think you alluded to that as well in your presentation. The question that sorry.
So if you may just add a small comment to what has been said on which kind of expert, I would also outline that in IMR is also very important to evaluate what would be, and that would be the scope of marketing competitive intelligence, to identify what would be the freedom to operate and what are the potential areas for innovation. And I would also add that critical, I would say, it's also critical to get the input of key opinion leaders, especially if new modalities or new route of administration are envisaged, because at the end, the ultimate user of a new product will be the clinician and patient. And if they're not ready to take a new antibiotic, it will be a failure of the antibiotic itself. So this is also something that should be really taken into account since the beginning, if new avenues want to be need to be explored. Sorry for making the point, but it's something that I think is really relevant for IMR nowadays.
Absolutely, fully agree with you. Absolutely. Different question. Yeah. This is a bit more tricky, I think. Is there any difference in the target product profile for a best-in-class or a first-in-class antibiotic? So okay, maybe Francesca, you can start.
Yeah, that could be indeed a broad topic. I would say from my point of view, to develop a TPP for a best-in-class may be slightly easier, because in this case, you would compare the activity of a new antibiotics to something which is already existing in the market. So the selection criteria could be based, of the new drug could be based on what is available from the other drugs, and the clinical path is well defined for a best-in-class. So the TPP would mainly address points like avoiding cross-resistance with the antibiotics of the same class being already in the market. So I would see that as the main critical point. For the rest, the toxicity and efficacy parameters should be relatively easy to be set up, because again, there you have some comparison. First-in-class antibiotic could mean several different things. You may have a new scaffold addressing an old target. You may have an antibiotic with completely new mode of action, a completely new modality. So in that case, let's say what could be particularly difficult to identify a selection criteria could be the toxicology and the efficacy for the new approach. And again, I reiterate the message again, then the clinical path may also represent a challenge. So in that case, that would be for sure an additional challenge for a best-in-class. Sorry, for a first-in-class, sorry, excuse me.
Yeah, Francesca, just to remember, there was also a similar webinar and a similar discussion panel in the AMR conference with Malcolm Page and Roche. I think they talk also about the compound classes or different series that you have, and about target candidate profiles. And then I think you will have multiple TCPs, target candidate profiles, linked to the same TPP. So that's also an element of when you mention different compound classes to choose from, right? So we would have probably also developed TCPs on that, right? I think you are doing this, huh?
Yeah, let's say I would consider the PPP as the frame describing the type of infection you want to treat, the population, and so on and so forth. And then the TCP would really give which are the characteristics of the new antibiotics that will enable you to treat this kind of infection. So and then indeed, if you consider that different bacteria can be approached or tackled by different approaches, then of course you can have more than one TCP associated to one TPP. However, it depends on the size of the company. Of course, you cannot eventually pursue any kind of TCP for the same TPP, and would rather focus on 1, 2, eventually 3 different TCPs. But this is my personal opinion. I don't know if my colleagues here have a different view.
Okay, keep it. We have a different, I'm just jumping a bit from topic to topic, but so also an interesting question, by the way, is to what extent do you do the three of you or all three involve patients in the development of TPPs for antibiotics? When and how do you involve them? This is a question from Christina Erling from Ligature. Maybe Gareth, you can start. Yeah, I think you already mentioned you mentioned in your presentation a little bit, but please elaborate a bit.
Yeah, it's a great question, and we do in GSK. I mean, throughout GSK, we try to engage with patients or patient groups around all aspects of our development, and obviously the TPP is a key area of that. We need to really understand what the patient needs and what the patient needs are. So getting that patient voice in to understanding that is absolutely crucial. we take that beyond even into trying to get the patient voice into the way that we design our clinical trials, but clearly getting that up front in terms of understanding from the patient themselves what are the unmet needs for you, what are the problems with the current treatments. If someone was developing a perfect drug, what would it need to look like, and how would it need to be better than what you're taking now, is crucial input to developing that TPP.
Erin, you want to add something to this?
Yeah, I mean, I think we're getting better, frankly, at when and how to do this. So we've nucleated advisories, which include patient voices for specific areas. I'll give an example, which would be cystic fibrosis as an example, to understand, again, from the patient perspective, especially because these people are on, lots of meds for a long time, what they really feel would be impactful for them. Part of it is framed with, again, at different levels of the healthcare system, what people need. But I still think it's important to say that there's a lot more we could do to be effective in the patient voice area.
Thank you very much. There's plenty of more questions for you guys, but I don't want to sort of overwhelm you, but I'm just trying to see which one could be the most relevant. Maybe we have one or two more questions before we have to end the session, but I'm going to go back to Gareth, if you don't mind, but it's relevant for the three of you. So you have now your TPP, you developed it, maybe you have different types of TPPs. How do you keep the TPP current and relevant? So you will have new results coming in from the clinical development, or changes in the external landscape, new guidelines that come up, competitors that are popping up. So yeah, I think it's relevant for the three of you, but maybe Gareth, you can start. How do you keep your TPP current and relevant?
Yeah, no, really great question, and really important, and agree with the comment Erin made that it's, it is a living document, and it has to be, because things change. Things change with respect to what we know about the drug we're developing, but things change outside as well, and they change the disease space. There could be a newly licensed drug, there could be some change in guidelines, some significant change in the way that a disease is handled, a new diagnostic might appear, and we have to adapt to those things, because developing a drug takes years, and things will happen during that period of time. And of course we try to future-proof the best we can, but you can't predict everything. So, I mean, key points when we need to look at that are obviously when we generate new data. So we would look at those primarily, are those data meeting with our TPP or not? And then we have a couple of questions, which is, does that mean we should stop? Or actually, is there an adaptation of this TPP that means there's still a path forward for this drug that meets an unmet need for some patient group somewhere? And if that's the case, then we would make an adaptation to that TPP. We would see governance endorsement about changing the direction that we're, governance endorsement inside the company about changing the direction of our travel with that particular drug, and we would, obviously document and record any changes that we make to the TPP. And then the other aspect is the changes in the external space, and that might mean that, in particular, most likely, that might mean that the bar has now been raised in terms of your TPP. You might have thought a certain level of efficacy or safety or requirements would have been sufficient to get your drug out there and access to patients, and now the bar's been moved upwards, and then you need to adjust your TPP accordingly, and obviously reevaluate where you're at, and if you can get there, possibly make adaptations to your downstream development plan to generate the right evidence.
Thank you, Gareth. Thank you very much. Erin, you want to add?
Yeah, so I think Gareth really described a lot of the same activities, very well. One thing I'll say, everybody knows that we fund in stages. And in order to progress from one stage to the next, we do nucleated advisory board, and it's kind of an uncomfortable thing, because it's not just about milestone achievements. So have you check the box from sort of A to E or whatever your milestone numbers are happen to be, but rather, do you remain relevant in the face of all those changes that you highlighted? And this can be painful and tricky, but I think we all have to keep ourselves honest in order to bring, the best products we can.
Thank you. I'm very cautious of the time, and I want to finish with something positive from all of you, from the three of you. Any final piece of advice? And I know there is on the websites and on CARB-X there's a lot of guidelines, but just from a personal note, because I don't want people now go home and be very desperate about TPPs and they're very complex and they're never gonna be achieving. So can you just give some final recommendations from Evotec and GSK and CARB-X in a positive way, if you can? Francesca, you can start. Any final advice to the audience?
I would say that the take-home message from my showcase is that I should recommend, let yourself to be positively surprised.
Thank you.
If you are not able, or let's say if you do not have this kind of attitude, maybe it's not the right area to tackle.
It's probably true for every researcher in general, but it's a very good point. Gareth, ending with a positive note.
Yeah, I think my positive note is that the TPP is your best friend in terms of navigating drug development. It really is. If you get that right and you define it right with the right inputs, it will guide you as to making the right decisions. So see it as a positive thing. Thank
you, Gareth. And Erin?
Yeah, I mean, I would say that the thing about the TPP is it provides some structure. Entrepreneurs are often not great about structure, and so you have to have that balance of really being out there and passionate when no one else is, but then understanding sort of where and how you need to stay within some bounds to to make the progress. So it's a balance, but it's a good balance.
All right. I'm glad that we end with this positive note, to be honest. So the only thing I can do is on behalf of the AMR Accelerator and my combined colleagues and the whole audience. I think at some point we were with 170 people or something. So really great. This is thanks to you, the speakers, and also thanks to people that are really interested in that topic. I'm sure that there needs to be a follow-up on this. And I'm sure there will be other organizations also talking about TPPs. I think there is an upcoming session from GARD-P as well, somewhere in April or May. So please check the Revive sessions as well. We will probably also have more. I'm sure that CARB-X will also have more communications on this, as this is a very important topic. We will, if you have more questions, because we haven't been able to address all the questions, there was basically too many. We'll follow up with you. And yeah, as a final note, thank you, Francesca and Erin and Gareth, for your contribution, for preparing it. And thanks a lot.
Thanks so much.
You're welcome.
Thanks to you for the organization.
Bye, everybody.